Hi, my name is Allison Martin, and I'm a biochemistry student at SUNY Fredonia. I'm here today to give some highlights from one of the posters I'll be presenting at this year's Fredonia Research Expo. This poster is a synopsis of the research I did from the summer and fall of 2013, and this is a population study of Lilaeopsis Shaffneriana subspecies recurva. Lilaeopsis Shaffneriana subspecies recurva is better known as the Huachuca water umbel. This plant is limited to ciénagas, which are rare marshy wetlands in the Sonoran Desert, and it needs shallow, slow-flowing waters. In these locations, it grows in large grassy mats, as you can see in the bottom image, and has tiny flowers, as pictured on the right. So why would we want to look at the Huachuca water umbel? This plant, along with two other species, was listed as endangered in 1997, and its habitat, the ciénagas, was federally listed as critical in 1999. HW is an indicator species for the ciénagas, and the loss of habitat has drastically reduced its range, such that there are only 12 extant populations known currently. Of these, 2, as well as a third that is now extinct in the wild, are cultivated in the Jewett greenhouse. In order to craft a reintroduction plan, a genetic population study was needed, which recently became possible to do as variable microsatellites were identified in 2012 by Tu et al. In doing this population study, I used nested PCR. Here we have 2 primers that are specific to the locus and a third which contains the fluorescent tag. So microsatellites are small sections of DNA with multiple repeats. Microsatellite loci are commonly used in identity testing, where variable numbers of repeats are used to identify an allele. Varied alleles will then give a specific genotype that can be used to identify an individual. Tu et al. Identified 8 loci for this specific to HWU. In doing this study, I used 3% agarose gels as well as polyacrylamide gels in a Lilaeopsis genetic analyzer to determine the exact base pair size of the alleles that we had in these populations. ImageJ was then used to analyze the gel images. Here we have some sample gels. On the right, we have 2 loci, LASC 9 and 39. 9 shows the variation, which is exactly what we were looking for. We have a heterozygote on the left and 2 different homozygotes in the middle and on the right. LASC 39, however, is not variate between the different populations. Once amplification was determined, I would then run the samples on the Lilaeopsis polyacrylamide and then analyze that image using ImageJ electropherograms. On the right is a sample of one of the electropherograms. As you can see, the red molecular weight ladder across the bottom and the green alleles in the middle. And this allowed me to determine the exact base pair size of our alleles. As these were new primers, it took quite a lot of troubleshooting and PCR optimization and eventually redesign of a couple of the primers to determine exactly what genotypes we had in the populations here. Unfortunately, while we were looking for variation, what we found was quite a lot of conserved loci. 6 of the 8 microsatellite loci were not variate between our different populations. We only had 2 points of variation at LASC 4 and LASC 9. This was not entirely unexpected, as it turned out that we had only a single individual from each population rather than multiple individuals as we had first thought. However, the implications for this are that without a lot of diversity in the plant, cultivation and reintroduction become much simpler, but the lack of diversity may not bode well for the future of the species. However, we cannot say this definitively since microsatellites have no phenotypic consequences. More work certainly needs to be done with a larger sample of populations, but unfortunately, due to distance and cost, I will not be able to do this. I'm currently focusing on Frasera caroliniensis, which is a local rare species, to develop similar molecular markers for population studies using rapid SCAR analysis. A quick note of thanks for funding and for advice and assistance to the following. And if you're interested, a free PDF of this poster can be downloaded at tinyurl.com/mtszb79. So thanks for watching, and I hope you enjoyed it.